was it? Oh my god, I'm gonna be late for school. Oh wait, I'm 29 years old. Oh, I'm a teacher! Made it just in time. Hello, Psych class. I hope you enjoyed my over-dramatic introduction. As you can probably already tell from these videos alone, I may have a little too much time on my hands. Anyways, welcome back to our virtual classroom for another AP unit review. Today we're going to be looking at Unit 2, the biological basis of behavior. In this unit review, we are going to start off by taking a look at the interaction between our heredity and the environment. Then we're going to take a quick look at the endocrine system and see what role it plays in regards to our behavior. From there, we will move on to the nervous system. Here we will take a look at the different divisions and parts of the nervous system. Our next step is going to be a tour of the brain, where we take a look at major brain areas and their functions. Then we are going to end things on my favorite topic, sleep and dreams. We're going to see why exactly we dream and what the importance of sleep is, as well as some other cool facts about sleeping and dreaming. Now, before we get started, remember to have your review guide in hand. While watching the video, be sure to fill out your review guide so you have all the information you need. If you need a review guide, you can click this link right here to my introduction video. In the description, you will find a link to a Google Drive where you can download the review guide. Remember, the more serious you take the review guide, the better you will score on the AP Psych test. Now let's get started. The biological basis of behavior is going to center around the biological perspective of psychology. Remember from our Unit 1 video review, the biological basis of psychology aims to explain behavior through physiological means. So mainly we're going to be taking a look at our nervous system and our genetics. When talking about the biological basis of psychology, it is important to have the mindset that everything psychological is biological. Whether it is sight or sound, a memory or an emotion, every one of our psychological experiences triggers some type of reaction in our brain and body. The reactions to these corresponding behaviors are what we are going to focus on during this unit review. So to what extent exactly is our behavior determined by our heredity? What about our environment and who we surround ourselves with? As you already know, this is the nature versus nurture controversy, which we briefly touched base upon in our Unit 1 review. When a healthy baby is conceived, it will have a total of 46 chromosomes, 23 from the mother and 23 from the father. Chromosomes are simply a structure in the nucleus of a cell that contain our genetics, which are determined by our DNA sequence. DNA, or deoxyribonucleic acid, is going to be a molecule that contains the genetic code for an organism. All living things, your dog, the annoying bug flying around your house, your parents' ficus outside, all have their own unique DNA. Your genes are just going to be a unit of heredity that are transferred from your parents to you and is held to determine some of your characteristics. Charles Darwin's research led to the creation of the evolutionary perspective of psychology, which focuses on how natural selection favored behaviors that contributed to survival and the spread of our ancestors' genes. Natural selection is just going to be the process where traits that make it more likely for an organism to survive will have a better chance to reproduce to the next generation. Mutation is going to occur when there is a change in DNA. If this mutation is a naturally selected trait, it will be passed on to the next generation and continue to be reproduced. So changes in an organism's DNA can cause changes in all aspects of life. Mutations are essential for evolution. They are the raw material of genetic variations. Without mutation, evolution could not occur. Today, behavior geneticists study the role played by our genes and environment in mental ability, emotional stability, temperament, personality, interests, and a whole bunch of different things. They look at the causes of our individual differences. So how exactly do behavior geneticists achieve this goal? Among other things, a very popular type of study to measure the influence of nature versus nurture has been identical twins. Since identical twins share the exact same DNA, they are the perfect test subjects to determine how genetics and our environment play a role in our development. While not the major bodily system in regards to behavior, it is very important to understand the basic workings of the endocrine system and its influence on our behavior. The endocrine system is the system in the body that regulates all of our body's hormones. A hormone is just going to be a chemical messenger that is released from the endocrine system into the bloodstream to influence the nervous system to regulate behaviors such as aggression or love. Here we have the hypothalamus, a small region of the brain located at the base of the brain. While the hypothalamus is very small, it plays a very crucial role in regulating the endocrine system. Neighboring the hypothalamus, we have the pituitary gland. The pituitary gland is considered the master gland because it controls the activity of most other hormone-secreting glands. Another tiny gland located in the brain is the pineal gland. Its function is not fully understood, but researchers do know that it does regulate some of our other hormones. Melatonin, which we'll talk about a little later, which is our sleep hormone, being one of them. The last gland we will mention is the adrenal gland. Whenever your body goes into fight or flight, this could be you playing a sport, getting scared while watching a horror movie, whatever, your adrenal glands will release adrenaline. The endocrine system does serve several other functions that I didn't really touch base upon, but for the most part, that's what you need to know about the endocrine system for AP Psych. The next system we are going to talk about is the nervous system, which is going to be the body's primary communication network. The nervous system can be divided into two sections: the central nervous system and the peripheral nervous system. The central nervous system contains the brain and the spinal cord and is the central location for all behaviors and mental processes. The brain works as the command center, while the spinal cord handles all the incoming and outgoing messages. Sometimes a message will bypass the brain and be sent straight from the spinal cord. We call these spinal reflexes. Imagine you walk into your kitchen to talk to someone in your family, and while you're getting into the conversation, you unknowingly put your hand down on a hot stove. Without even thinking, you'll pull your hand away to avoid injury. Instead of going through the normal process of your hand sending information to your brain, saying, ouch, this hurts, your spinal cord jumps in and has you move your hand right away. This happens very quickly with almost little to none conscious awareness. It's no different when you go to the doctors and they take that little mini tomahawk to your knee to check your reflexes. The peripheral nervous system is a subdivision of the nervous system that contains all of your nerve endings throughout your body. The peripheral nervous system has its own two subdivisions. The autonomic nervous system regulates involuntary functions such as breathing, heart rate, and digestion. The autonomic nervous system also has two subdivisions: the sympathetic nervous system and the parasympathetic nervous system. The sympathetic nervous system is activated during a period of fight or flight where our body perceives some type of threat. As I mentioned before, our fight or flight response is activated when we are in an emergency. It is an instinctive physiological response to a perceived threat. This shows us an example of how the endocrine system and the nervous system work together. When our fight or flight response is triggered, our hypothalamus is able to relay this information to our endocrine system, allowing for our adrenal glands to produce adrenaline. The parasympathetic nervous system, on the other hand, is responsible for bringing our body back to a normal state of functioning after experiencing the fight or flight response. Moving on to the second subdivision of the peripheral nervous system, which will be the somatic nervous system. The somatic nervous system is in charge of all of our voluntary functions. Things like walking to our car or grabbing a drink are all possible because of the somatic nervous system. Now let's dig a little bit deeper into the nervous system. Inside of your brain, you have billions of tiny cells called neurons, whose job it is to communicate information throughout your body. Brain tissue no larger than a grain of rice can contain up to 10,000 neurons. Every neuron has a glial cell, or several, since glial cells outnumber the neuron 10 to 1. The glial cell is much smaller than a neuron, but it does serve an important function of providing nutrition and removing waste, such as dead cells, so the neuron can properly function. When looking at the anatomy of a neuron, there are several parts that we need to know. The nucleus just contains the cell's genetic materials. The cell body, or soma, provides structural support for the cell and also assists in sending neural messages. Dendrites branch off of the cell body and are responsible for receiving messages. The myelin sheath is a fatty substance that surrounds the axon. It's going to be responsible for speeding up neural transmission speeds. The node of Ranvier is going to be the gap in between the myelin sheath. Having this gap allows for the message to diffuse in and out of the neuron. The way I like to think of the myelin sheath and the node of Ranvier working together is by taking a look at my old Hot Wheels toy, Crisscross Crash. In the center of this toy, they would have these little motors that would propel the car forward. Multiple motors allow for the car to keep speeding up. The motor could be considered the myelin sheath, while the areas in between these two motors could be considered the node of Ranvier. Man, now I need to call my mom to see if she still has my Hot Wheels collection. The axon is going to be the part of the neuron that is responsible for sending information to the next neuron. At the tip of each axon, we have our axon terminal. This is where chemical messengers known as neurotransmitters are stored in preparation of being sent. sent. Schwann cells are just the name of the cells that wrap around the axon to form the myelin sheath. Schwann cells are going to be a form of glial cells. So now that we have the anatomy of a neuron down, let's talk about the three different types of neurons. We are going to have afferent or sensory neurons, efferent or motor neurons, and interneurons. Sensory neurons are going to serve two important functions. One, to take information from the environment and report it back to the brain, so things such as light or sound for our hearing and vision. And two, to take information from our skin and internal organs to send back to our brain. So when you stub your toe or hit your shin on the end table, it's your sensory neurons letting your brain know to send the maximum pain threshold down to that area. Our motor neurons are going to be responsible for communicating information to the muscles and glands of the body. A simple act such as blinking your eyes activates over 1,000 motor neurons. Both sensory and motor neurons are going to be located in the peripheral nervous system. And last but not least, we have the interneuron, the number one neuron in terms of quantity. The interneuron is in charge of communicating information from neuron to neuron. The interneurons can be found in the brain and spinal cord, making it a part of the central nervous system. As I mentioned, the neuron is the primary communication cell in the body, so now we have to talk about how exactly these cells communicate with one another. When the axon of a presynaptic neuron meets the dendrites of a postsynaptic neuron, neural firing can begin. For a neuron to have sufficient energy to send a message, an action potential must occur. An action potential is just going to be a brief electrical impulse gathered by the cell body and the dendrites that is going to stimulate the cell just enough to allow for a message to be sent. For a neuron to have an action potential, it must receive enough stimulation to reach its stimulus threshold. This allows the neuron to have enough energy to depolarize and have an action potential. When a neuron receives enough stimulation, an action potential will always occur. The neuron will either receive enough stimulation for an action potential to occur, or it will not, and an action potential won't occur. Seems pretty obvious, but this is a principle referred to as the all or none law. After a neuron has had an action potential, it goes into a brief refractory period where it is basically recharging. During this time, the neuron cannot have another action potential. After the refractory period, the neuron returns to its resting state. The resting potential is just going to be when a neuron is ready to fire off another action potential if it receives enough stimulation. The axons of the presynaptic neuron and the dendrites of the postsynaptic neuron never actually touch. Instead, a tiny fluid-filled space is created in between the two. A little fun fact, a synaptic gap is roughly 20 to 40 nanometers in length. For comparison, a standard sheet of computer paper is roughly 100,000 nanometers in length. After a synaptic gap is formed, a neuron can then send its message. Neurotransmitters are going to be chemical messengers that are stored in tiny sacs in the axon terminal. These tiny sacs are called synaptic vesicles. These neurons then link up to a specific receptor site on the dendrites of the postsynaptic neuron. Each neurotransmitter has its own specific chemical makeup, allowing them to attach to their designated receptor site. The way I like to think of this is imagining one of those little kid toys, that little cube where they have the shapes cut out and the kid has to take the little toys and put the star in the star shape, the square in the square shape, and the circle in the circle shape. You get the point. After a neurotransmitter attaches to its receptor site, a message is communicated. Oftentimes, it will detach from the receptor site and be reabsorbed by the presynaptic neuron. This is a process known as reuptake, which plays a huge role in how psychotropic medications affect our behavior. There are over 100 neurotransmitters in the human body, but luckily for us, we are just going to take a look at 7 of them. Your ability to feel, think, see, react is all going to depend on the delicate balance of neurotransmitters in your nervous system. If this balance is altered, it can lead to physical and behavioral changes. Dopamine is going to be involved in movement, attention, learning, and the experience of pleasurable sensations. Studies have shown that the addictiveness of several drugs, such as nicotine and cocaine, is related to the substance's ability to increase dopamine activity in the brain. Parkinson's disease has been linked to the brain not producing enough dopamine, while too much dopamine can actually lead to hallucinations and perceptual distortions. One of the leading theories on the cause of schizophrenia is the dopamine hypothesis, which states that an excess of dopamine in the brain causes the symptoms to occur. Serotonin receptors are found in multiple areas of the brain. Serotonin is involved in sleep, sensory perception, moods, and your emotional state. A lack of serotonin in the brain can be linked to depression. SSRIs, or selective serotonin reuptake inhibitors, are a common form of prescribed antidepressants. These drugs block the reuptake of serotonin neurotransmitters, leaving them to flood your synaptic gap in order to combat the symptoms of depression. Norepinephrine is going to play a key role in our fight or flight response while regulating sleep, memory, and learning. An excess of norepinephrine can be associated with anxiety. Gamma-aminobutyric acid, aka GABA, works to slow down brain activity. Too much GABA impairs learning and motivation, while too little can lead to seizures. When someone drinks alcohol, it increases the amount of GABA in the brain, leading to the feelings of relaxation. If an individual suffering from alcoholism decides they want to stop drinking, this can actually be very dangerous. The individual can experience seizures, or in extreme cases, even death, since the continual alcohol use messes with the production of GABA in the brain. Acetylcholine was the first neurotransmitter ever discovered. Found in all of our motor neurons, it is acetylcholine's duty to stimulate our muscles to contract, as well as helping with our learning and memory. A disease associated with the lack of acetylcholine production, endorphins are our body's natural painkillers. Similar to that in structure of morphine and other opioids, endorphins are hundreds of times more potent and are released in response to stress, trauma, and pain. If you've ever been on a run and experienced a runner's high before, this is just your synaptic gap being flooded with endorphins during your run, giving you that nice feeling. Our last neurotransmitter that we are going to talk about is going to be glutamate. Glutamate counteracts GABA and speeds up brain activity. Glutamate is also involved in learning, memory, and sensory processing. Excessive glutamate can excite nerve cells to their death, which is a process known as excitotoxicity. Neurotransmitters can either be inhibitory or excitatory. Inhibitory neurotransmitters work to slow down brain activity, while excitatory neurotransmitters work to speed it up. While most neurotransmitters are either excitatory or inhibitory, some, such as dopamine, can be both depending on which area of the brain it is located. Psychoactive substances can also play a huge role in how our neurotransmitters operate. An agonist is going to be a substance or drug that binds to a receptor site and triggers a response in a cell. As I mentioned earlier, alcohol mimics the effects of GABA, meaning it is a GABA agonist. When alcohol is consumed, the alcohol binds to the GABA receptor site, therefore stimulating the cell to produce an effect. On the other hand, an antagonist is going to be a drug or a substance that blocks the effects of a neurotransmitter. When there's an opioid overdose, the drug naloxone, more commonly referred to as Narcan, fights the opiates in the brain for a spot on the opiate receptor, causing for a reversal of the effects of the drug. For our next topic of discussion, we are going to talk all about the human brain. Since we are in the virtual world, we are going to take a little trip inside the old dome for a first-hand look at the human brain. Alright, so here we are inside the human head. We've got our brain over here. Let's get started. Strap in, because this is going to be very quick. Smells kind of funky in here. Now, there are several areas of the brain that you will find online or in your textbook, but I'm just going to go over the ones that I feel are the most important for the AP Psych test. The first area of the brain we are going to focus on is going to be the brainstem. The brainstem connects the spinal cord to the rest of the brain and will hold more of the primitive brain structures that regulate vital life functions. The main responsibility of the medulla is going to be to control vital life functions such as breathing and heart rate. The pons is going to help coordinate movements on the left and right side of our body. The cerebellum coordinates movement, balance, and posture, and the reticular formation helps regulate attention and alertness. Also part of the brainstem, but located in the midbrain rather than the hindbrain, is going to be the basal ganglia. The basal ganglia is going to contain the substantia nigra, which is the major part of the brain responsible for dopamine production. Our cerebral cortex, located on the forebrain, is going to have 4 pairs of major lobes, meaning there is going to be a lobe on both the left and right hemisphere of the brain. Also part of the cerebral cortex is going to be your corpus callosum. This is the bridge that links your left and right cerebral hemispheres in order for them to communicate with one another. The temporal lobe, located above our ear, is responsible for processing auditory information. The occipital lobe in the back of our head allows us to process visual information. The parietal lobe is going to process somatic sensations, so these are things such as hot or cold or pressure on your hand. The frontal lobe, located at the front of our cerebral cortex, is going to be by far the largest lobes in the brain. The frontal lobe processes voluntary muscle movement and is involved in thinking, planning, and emotional control. Another area of the forebrain is the limbic system, which are brain areas that border around the brain stem. The hippocampus is a large structure in the temporal lobe of both cerebral hemispheres. The hippocampus is going to play a huge role in learning and the consolidation of our newly formed memories. You will always remember if you see a hippocampus. The thalamus processes and integrates all sensory memories except for smell and sends them to the cerebral cortex. The hypothalamus links the brain to the endocrine system while regulating hunger, thirst, sleep, and sexual behavior. And last, for the limbic system, we have the amygdala. The amygdala is a very small, primitive area of the brain responsible for memory and emotion, especially fear and anger. The last areas of the brain we talk about are going to be the cortical areas of the brain. These cortical areas are just specialized areas located in the cerebral cortex. The prefrontal lobe, located in the frontal cortex, is responsible for problem solving, emotional control, and complex thought. The primary somatosensory cortex is a cortical area in the brain located in the parietal lobe. Its job is to process somatic sensations throughout the body, such as proprioception, touch, pain, and temperature. The primary motor cortex, located in the frontal lobe, is responsible in generating neural impulses that control the execution of movement. The next two cortical areas have to do with the comprehension and processing of language. In the 1860s, neuroscientist Paul Broca discovered that if an area in the brain located in the lower left part of the frontal lobe is damaged, it would cause issues with the ability to produce speech. Broca's area is the part of the brain responsible for productive speech. A decade after Broca's discovery, German neuroscientist Karl Wernicke discovered another important area of the brain in regards to our language. Located in between the parietal and temporal lobe, Wernicke's area is responsible for our ability to comprehend language or our receptive speech. When there's damage in either Broca or Wernicke's area, it results in aphasia, or the partial or complete inability to articulate ideas or understand spoken or written language. The idea that different functions of the brain are located or localized in different areas of the brain is an idea known as cortical localization, while lateralization of function refers to the specific notion that psychological or cognitive functions are processed primarily on one side of the brain. To further understand this idea, we are going to talk about the split brain patient. But first, let's get out of this head and head back to the classroom. Alright, I hope we all made it back safely. Let's get started talking about the split brain patient. The split brain operation is a surgical procedure where the corpus callosum is cut. This severs the connection between the left hemisphere and the right hemisphere of the cerebral cortex. So why exactly would someone want to receive this procedure? As I mentioned earlier, the corpus callosum is like the bridge that connects the two sides of the brain. In a normally functioning brain, it can be seen as a well-constructed bridge. You have multiple lanes going both ways, separate traffic in order to get from one side to the other. Well, I want you to imagine that this bridge only had one lane and everyone driving through it was blindfolded. Well, you can imagine the amount of chaos that would unfold if that were the case. When neurons fire in a disorganized fashion across the corpus callosum, it can lead to the individual experiencing epileptic seizures. Severing this tie dramatically decreases and sometimes even completely removes these seizures. However, this does lead to our two cerebral hemispheres now operating independent from one another. When first conducted in the 1940s, there appeared to be no negative side effects as a result of severing this connection. A famous psychologist known as Carl Lashley even joked about the function of the corpus callosum, stating that its main purpose was to keep the two cerebral hemispheres from sagging down. It was not until the 1960s when Roger Sperry and his colleagues started to unravel the puzzle of the left and right hemispheres. To test the abilities of the split brain patient, Sperry would use an apparatus similar to what is shown on the board. Sperry would have the subject stare at a focal point on the screen and flash a word or an image to either the left or the right of that focal point. Stimuli on the left was processed by the right hemisphere, while stimuli on the right was processed by the left hemisphere. When the right side of the screen flashed the word face, the sensory information is brought in through the right eye and taken to the left hemisphere of the brain. When asked to recall what was seen on the screen, the subject would easily be able to say that they saw the word face. This is because the information was sent to the verbal left hemisphere. Remember, we just talked about how Broca and Wernicke's area are located on the left hemisphere. Now this is where things get interesting. What would happen if the same word was projected on the left side of the screen? In this situation, the information would go through the left eye and be sent to the nonverbal right hemisphere. When asked to recall what the subject saw, they would say nothing. That's because all verbal information is going to be processed in the left hemisphere. Since the information went to the right hemisphere and the corpus callosum has been severed, the individual has no way of processing the verbal information because the left hemisphere can no longer communicate with the right. However, this information still is being processed by the right hemisphere. When asked to draw a picture with their left hand, the subject would draw a face, because that's the word that they saw, showing nonverbal comprehension of the presented stimuli. Michael Gazzaniga, a psychologist who worked with Sperry, continued split brain research, specifically focusing on lateralization of function. Rather than me going on and on about Gazzaniga's work, I'm just going to show you a quick video clip of his experiment.
Instead of naming the word, I want you to point to the word.
Again, Joe sees two words simultaneously. Bell goes to his non-speaking right brain. Music to his speaking left brain. When asked to point to a picture of what he saw, he chooses Bell. But when asked why...
Okay, why'd you pick that one?
Music.
Music?
And when asked to explain...
There was music and bell, and those few minutes ago, last time I heard any music was coming from the bells out here.
Uh-huh.
Banging away.
So the bell's outside here?
So... Look, good enough answer to me.
What's extraordinary is that Joe's speaking left brain concocts a plausible story of why he pointed to Bell, even when some of the other pictures more obviously represent music. Gazzaniga believes this determination to find cause and effect, this desire to explain, is the left hemisphere's most marvelous property.
One of the unique things to the human brain is this need to interpret why two events occurred. What was the antecedent to this? What caused this? And if you can imagine that a species like us that has that little chip in its brain that asks those questions is going to survive rather well, because it's going to figure out more about the nature of the world than a species that doesn't have that.
Okay, Joe, I want you to keep your hand.
Our next topic in regards to the human brain is going to be brain scans. With our 21st century technology, we have multiple methods that allow us to scan the human brain. One of the most well-known brain scans out there is going to be the magnetic resonance imaging, or MRI for short. An MRI machine works by using very, very strong magnets. I'm talking about 1,000 times stronger than your normal fridge magnet. The magnetic fields in the MRI cause the proteins in the body to align with one another. Radio waves are then pulsed through the patient, stimulating the protons and allowing for an imaging capture device to detect internal organs. The result of an MRI scan are clear, detailed pictures of internal organs. Images are received in 2D cross-sectional slices that can be combined to form a 3D image. The F in fMRI stands for functional. The fMRI operates under the same principles of an MRI, but our results are going to be a little different. The fMRI measures small changes in blood flow that occur in the brain over time. These scans show us which part of the brain is handling different functions. fMRIs are generally used more in a research setting, while MRIs are used mainly for health reasons. Our next brain scan is going to be the computerized axial tomography, or also known as a CAT scan, sometimes referred to as a CT scan. During the CAT scan, the individual is placed on a bed that goes through a tube. The tube shoots narrow beams of X-rays through the patient's body. The digital X-ray detectors then produce 2D images. These 2D images can then be combined in a computer to produce a 3D model of the brain. Next, we have the positron emission tomography, or PET scan. During a PET scan, the patient is injected with a very small amount of radioactive glucose, which acts as a tracer substance. The scan is then able to detect the traces of radioactivity from outside the scalp. This allows for the brain scan to detect any type of metabolic activity or blood flow within the brain. A computer can then use this absorption data to show levels of brain activity in a color-coded map. Red equals high brain activity, while blue equals low brain activity. The last brain scan that we are going to talk about, which just so happens to be the first brain scan ever invented, is going to be the electroencephalography, or EEG for short. With the EEG, you have small electrodes placed on a cap that the patient wears to measure brain waves. These electrodes then pick up electric activity that the brain naturally produces. Pens hooked up to the EEG then trace this brain activity onto a piece of paper. While it does not recreate an image of the brain, EEGs can provide us with a good amount of information, such as someone's state of consciousness or how long it takes to process various stimuli. Prior to brain scans, the only way that researchers were able to study the physical brain was through autopsy. This would be when the actual brain was removed from someone's head, post-mortem of course, in order to be studied. Another quick thing I'm going to throw in, prior to the introduction of psychotropic drugs, neurosurgeons would often use the practice of lesioning to treat patients with severe movement disorders. This is when specific areas of the brain were purposely damaged to target cells that control movement. The idea is that targeting these cells will lower the symptoms associated with the movement disorder. Lesioning was very popular in the 1960s, but in our modern time, medicine is generally the preferred course of action. However, in rare cases where patients do not respond to psychotropic medications, lesioning can still be used as a last resort. Among other incredible feats that our brain can achieve, we have the idea of neuroplasticity. This is just going to be your brain's ability to reorganize itself by forming new neural connections. Structural plasticity refers to the brain's ability to change its physical structure as a result of learning. So while you are watching this video and recording information on your review guide, your brain is making brand new synaptic connections to compensate for your learning. Functional plasticity, on the other hand, is going to be your brain's ability to shift functions from damaged area of the brains to healthy areas of the brain. For this, let's talk about a quick case study. In 1848, Phineas Gage, a kind and dependable railroad foreman, was blasting rock in Cavendish, Vermont, in order to lay down railroad tracks. For his job, Gage would use a tool known as a tamping rod in order to pack down the gunpowder. While working, some of Gage's crew members distracted him, and he completely forgot to put a layer of sand over top the gunpowder. As he struck his tamping rod into the hole filled with gunpowder, the gunpowder ignited, causing the tamping rod to go up through Gage's cheek, through his eye, through his frontal lobe. The rod, measuring 3 feet 8 inches and weighing about 13 pounds, was found 50 yards away from the blast. Gage was conscious the entire time. The rod damaged Gage's frontal lobe as well as severed the connection between the limbic system and the frontal lobe. After the accident, Gage was a different man. With rapidly changing emotions, his friends could hardly recognize him. Gage would be one of the first individuals for whom we've learned something about the connection between personality and different parts of our brain. I think it's pretty interesting to think about that during this whole entire time, after suffering such a traumatic brain injury, Gage's brain was still attempting to compensate for all the damage that was done to it. Through functional plasticity, the brain would try and take away some of the duties of the frontal lobe and bring it elsewhere. And for our last topic of discussion, we have our states of consciousness. Consciousness just refers to our personal awareness of mental activities, internal senses, and the external environment. There are going to be 3 levels of consciousness that we need to know about. The preconscious is information that we are not actively aware of, but we can easily become aware of at any given time. If I were to ask you to tell me what you had for breakfast today, you would easily be able to bring that information from your preconscious into your conscious awareness. The next level of conscious awareness is going to be the non-conscious level. The non-conscious is a level of conscious awareness devoted to processes completely inaccessible to consciousness. These are things such as blood flow, the secretion of hormones, and the lower level processing of sensations. And our third and final level of consciousness is going to be unconscious. Pioneered by Sigmund Freud, the unconscious mind is considered to be the level of consciousness that includes all of our repressed wishes and desires that are not directly available for conscious awareness. According to Sigmund Freud and other psychoanalysts, the unconscious mind plays a huge role in our behavior. Now, before we move on to my favorite topic of sleep and dreams, I want to do a little bit of backtracking and talk a little bit more about how psychoactive drugs affect our brain. We talk about neurotransmitters and how slight changes of these tiny chemicals can lead to interesting changes in human behavior. Well, now we're going to see how these psychoactive drugs influence our neurotransmitters, which in turn lead to changes in behavior. A psychoactive drug is just going to be any chemical substance that changes brain function and results in the alteration of perceptions, mood, consciousness, cognition, or behavior. Depressants are going to be a category of psychoactive drugs that depress or inhibit brain activity. These are drugs such as alcohol or benzodiazepines, which is a category that the drug Xanax falls under. Opiates such as morphine and heroin also can fall under the category of depressant. Stimulants serve an opposite purpose. Stimulants are a category of psychoactive drugs that increase brain activity, arouse behavior, and increase mental alertness. These are drugs such as caffeine, nicotine, and cocaine. The last category of psychoactive drugs that we are going to talk about is going to be hallucinogens. Hallucinogens are going to be a category of psychoactive drugs that distort perception and evoke vivid images in the absence of sensory stimuli. Examples of hallucinogenic drugs include lysergic acid dethylamide, street name LSD, psilocybin, which is the psychoactive component of mushrooms, and even tetrahydrocannabinol, which is the psychoactive ingredient found in cannabis, which, yes, is considered to be a mild hallucinogenic. Alright, now that the drug lecture is over, we can move on to sleep and dreams. Scientists theorize two major reasons for why we sleep: the restorative theory and the adaptive theory. The restorative theory is exactly what it sounds like. It states that we sleep so we can repair both our body and brain. This theory states that if we did not receive sufficient amount of sleep, our body would eventually break down. The adaptive theory basically states that we sleep at night because at night we are more vulnerable to predators. This kept our caveman ancestors out of harm's way. In our brain, there is also a small bundle of nerves known as the suprachiasmatic nucleus, or the SCN for short. The SCN is located in our hypothalamus and is responsible for controlling our circadian rhythm. What is the circadian rhythm exactly? Well, I am glad you asked. The circadian rhythm is our own internal 24-hour biological clock that works as our sleep-wake cycle. As nighttime approaches, our pineal gland produces my favorite hormone, melatonin. Melatonin is just going to be the hormone that helps regulate our sleep-wake cycle. I, for one, am a huge fan of Ollie's sleep melatonin gummies. They knock me right out. So you just put your phone down after telling yourself you were going to go to bed 2 hours ago and decide it's finally time for some sleep. Well, what exactly is going on in our heads while this is happening? Due to technology such as the EEG that we talked about earlier, we are able to see exactly what is happening. According to sleep scientists, we have two major stages of sleep: REM and NREM. NREM sleep can be characterized into three separate stages. During stage 1 of NREM sleep, our alpha brain waves start to turn to theta brain waves, which basically just means our brain is starting to slow down. This is when we start to disengage from all outside stimuli. This stage is just the onset of sleep, and it only lasts for a few minutes. Oftentimes during this stage, you can experience what is known as a hypnagogic hallucination. These are vivid sensory phenomena that occur during the onset of sleep. True story. One time I was about to enjoy a nice, peaceful slumber on my delightful Tempur-Pedic mattress, when out of nowhere I heard the loudest explosion ever. Naturally, I did the smartest thing I could think of. I went right to my computer and got onto WebMD. After first being told that I was either terminally ill or in a coma, I came across something called exploding head syndrome, or EHS, which, yes, it is a real thing. Exploding head syndrome is just a condition when someone hears very loud noises on the onset of sleep or when you're about to wake up. Terrifying stuff. Anyways, stage 2 NREM sleep is going to be characterized by two types of brain waves: sleep spindles and K-complex. During stage 2, your brain waves continue to decrease, and we start to see the emergence of delta brain waves. If you've ever woken yourself up from your arm or your leg twitching during sleep, chances are you were in stage 2 NREM sleep. Our last stage of NREM sleep is stage 3, which is just defined as a majority of our brain waves being characterized as delta brain waves. During this stage, all outside stimuli is cut off. And the final stage of sleep is called REM sleep, which stands for rapid eye movement. This is because during REM sleep, our eyes are constantly darting back and forth. Our visual and motor neurons are active just as they are in wakefulness, and this is the stage of sleep where dreams occur. On rare occasions, your mind regains consciousness before REM sleep is over. In this state, you might not be able to move any part of your body and might experience a couple freaky hypnagogic hallucinations. This is known as sleep paralysis. A normal sleep cycle lasts around 90 minutes, and if you're getting the required 8 hours of sleep, chances are you'll go through about 5 sleep cycles each night. REM sleep generally happens at the end of each cycle. Some of you may be thinking, hey, one time I came home from school, took a 20-minute nap, and I swear to you that I had a dream during that time. What's up with that? Well, if you're sleep deprived, like myself, you may experience what is known as REM rebound. REM rebound occurs when your body jump-starts the REM stage of sleep in order to compensate for lack of sleep. Oftentimes, it is not necessarily our fault that we cannot get enough sleep. According to the American Sleep Association, roughly 50 to 70 million Americans suffer from some type of sleep disorder. That is roughly 25% of the US population. Among our most common sleep disorder, we have insomnia. Insomnia is just when a person regularly experiences the inability to go asleep, trouble staying asleep, or they just don't feel adequately rested in the morning. Insomnia can be acute when it lasts for 1 day to a few weeks, but it can also be chronic, and that's when it lasts for 3 or more days, a week, for several months. On the reverse end of insomnia, we have narcolepsy, which is going to be one of the more rare sleep disorders. Narcolepsy is characterized by excessive daytime sleepiness. Those suffering from narcolepsy may experience brief episodes of involuntary sleep cycles, and in extreme cases, even hallucinations. Sleep apnea is characterized by a person repeatedly stopping breathing during sleep. The lack of breath leads to an interruption in the sleep cycle, making it hard for the body to get sufficient REM sleep. Sleep apnea is often associated with an unhealthy weight. As the soft tissue around the mouth and neck become more relaxed during sleep, they tend to sag down and block the airway. It is estimated that roughly 22 million Americans suffer from sleep apnea. Night terrors are characterized by intense fear and panic, frightening hallucinations, and no recall of the events when awoken. It's very important to be able to differentiate between nightmares and night terrors. Nightmares are just going to be unpleasant dreams that occur during REM sleep, while night terrors actually occur during stage 3 of NREM sleep. Around 6% of children usually experience night terrors, but luckily they'll eventually grow out of it. REM behavior disorder is going to be when an individual acts out their dreams. Caused by the brain's inability to suppress voluntary muscle movements, this disorder is generally seen in males over the age of 50. So these sleep disorders oftentimes lead to sleep deprivation. Sleep deprivation just occurs when someone consistently fails to get enough sleep every night. Sleep deprivation can have both psychological and physical effects. Physical symptoms include a weakened immune system, a risk of heart disease, high blood pressure, and even weight gain. Psychological symptoms can include trouble thinking and concentrating, mood changes, and memory issues. Whether you're being chased by an axe-wielding murderer while running incredibly slow, or you're at work or school, then you wake up just to have to go to work or school, yeah, that one happens to me a lot. Dreams are just going to be the thoughts, images, and sensations that occur in our head while we are asleep. In times of ancient civilizations such as the Greeks and Egyptians, dreams were considered supernatural occurrences or means of divine intervention. More recently, we have different ways explaining why dreams occur. We are going to focus on 2 different explanations. The activation synthesis model of dreaming explains dreams as a biological process. During sleep, our limbic system becomes more active while our frontal lobe becomes less active. Our brain synthesizes and interprets this random neural activity and attempts to make sense of it. Therefore, we have our dreams. Sigmund Freud considered dreams to be the royal road to the unconscious mind. Freud believed that when we go to sleep, our ego defense mechanisms are lowered and repressed materials come to awareness in a distorted form, this being our dream. Freud categorized dreams into 2 different types of content. Manifest content is considered the part of the dream that we remember, while latent content is the hidden meaning behind the dream. So let's say you go to bed one night and all of a sudden you're 50,000 feet above the earth, just plummeting down to the ground. Just before making contact, you wake up in a panic. Curious as to why you had this dream, you decide to do a little bit of research. Through your research, you come to the conclusion that you had that dream because there's an aspect in your life where you feel like you are losing control. The dream of you falling would be considered the manifest content, while the meaning of you losing control could be considered the latent content. Freud believed that identifying and understanding the latent content in our dreams is key to understanding our inner conflicts. Whew, my goodness, that was a lot of information. I'm just gonna have to see you guys next time for our Unit 3 review. I am out of here.